So, with that good start, yeah, so many thanks for this invitation. It's great fun. Don't worry, it's fine, it's great. And more than compensated by the reception and the warmth of the... So I'm following two very interesting and inspirational and luminary speakers, and they've introduced several concepts. And I'm coming in at Bart's kind of cellular phase of neurodegeneration, and also speaking a little bit to the concept of homeostasis in a broad sense. So I work on neurodegeneration. I'm also a clinician. I'm also a neurologist that sees dementia patients. So this is a very vivid and meaningful area for me. And Bart has talked to you about Alzheimer's, but there's a spectrum of these disorders, and this doesn't work. Ina, do you have your pointer?
I use this one.
No, it doesn't work.
Yeah, it's work.
No, that's mine that worked.
It's all right, I use yours. Okay. So, and as Bart said, by 2040, in fact, not even the second commonest cause of death, probably the commonest cause of death, and the world is aging. So South America's aging and China's aging, and this is a huge problem. And Bart represents the UK's interest in this by heading up a new £250 million institute and investment in dementia research to find a cause, because David Cameron, in his wisdom in 2012 at our Dementia G8 summit, promised a cure by 2025. So that's 7 years away. So all of these disorders, although they all have specific pathologies and specific misfolded proteins, have some common features. The commonest of which is the earliest loss of synapses. This is the earliest stage that's associated with cognitive failure, with memory loss, with the earliest symptoms and signs in all of these diseases is the loss of synapses, which are essential not just for encoding memory, but also for their trophic support to neurons. And then the second common feature is neuronal loss. Here you can see a completely degenerated hippocampus and this misfolded protein accumulation, whether it be A beta or tau or prion protein or TDP43, there's misfolded protein and neuronal loss following synaptic loss in all of these diseases. So the way I represent this for any disease is with these two curves. The decline in synapse number, the accumulation of misfolded protein. And this is really the critical determinant of what happens next. Because, as I said, there's a lot of redundancy in synapse number and synapse function. And cognitive symptoms emerge at about 50 to 60% loss of synapses. But then you only have another 10% to lose before you lose neurons. These pathways kick in as well, but this is the critical window for rescue and reversibility, because you can't replace lost neurons effectively, but you can repair degenerated synapses and disturbed networks. So I'm interested in the common mechanisms underlying these processes, and because potentially these could lead to common cures. So this is almost aiming at the generic cellular downstream effects of specific gene effects or specific loss of homeostatic mechanisms, and finding a common way of at least providing the first layer of treatment in this disorder, in these disorders. So how do we study these mechanisms? And Bart has mentioned the problem of the mouse models. So the mouse model we've focused it on is this model, which is not a model, it is the disease. So prion mice are not phenocopying a human disorder, they're not overexpressing particular human mutants in APP. These have prion disease, which completely phenocopies biochemically, clinically, pathologically, and in their transmission characteristics, the features of human and mammalian prion disorders. And so using these mice, we've studied the link between protein misfolding and neuronal cell death in a number of, and we've focused on two processes. One is the effects of misfolded protein accumulation. We've done a lot of work on the effects on the activation of the unfolded protein response. This is again speaks to dysregulated homeostasis, where misfolded proteins activate the downstream proteostatic pathway, which fails, and the sustained translational failure in the context of failing synapses and diseased neurons leads to neurodegeneration. And by either genetically or biochemically, in a number of different drugs, pathways, and different mouse models, be it of prion or tauopathy, or and in Alzheimer's models as well, and not our labs, but also Eric Klein's labs and now Sonnenberg's labs have all shown that modulating this pathway is is a promising pathway towards preventing neurodegeneration, and we're right through to repurposed drugs on this, which are ready for clinical trials and are in press at the moment. But what I want to talk to you today is this side of things, because without the declining synapse number, and I think Bart's curves about the A-beta levels speak to the same principle, none of these have the impact if you have a high synapse number. So if you change the slope of that curve, you change the onset of neuronal loss. And that, you don't need much of a change in that to change this. If you equate this to going to hospital and institutionalization for dementia, if you change that from 75 to 85, you've already made an enormous impact on global health, global economy, and human suffering. And in fact, most of my patients come to clinic with a synapse number of around here and some memory problems and some early cognitive problems. And if I could hold them there, or even reverse a little bit, their outcome would be entirely different. Their outlook for themselves, their families, etc., would be entirely different. So I want to talk about how we approached this problem, which is that synapses are going down in these disorders at the earliest phases for unknown reasons. And a sort of generic representation of that, this is synapse number in the hippocampus, and this is time, and this is when you get symptoms and UPR activation, but you can see that synapse numbers are critically compromised well before that. And I've avoided a timescale because it's for any model you want. And this is the critical time point. So what's causing this loss of synapses? So we thought about the concept of structural plasticity. So the generation and pruning of synapses is a crucial process going on all the time in all of us. It's obviously huge in development, but it's a major part of repair and response to injury, and it's an ongoing part of memory. So we're all, as Eric Kandel pointed out, we'll all come out with different brains at the end of this lecture because we'll have generated and made some synapses and pruned some others, and it's an ongoing process. And you can see that synapses might die early if there's an imbalance in this process, if plasticity is defective. In other words, if the regenerative drive is less effective than the pruning, or if the pruning drive is overactive, and here comes the microglia, etc., and these conversion pathways. But you can see that the effect of that would be net synapse loss. So what we hypothesized was that the reason synapses are dying across the spectrum in neurodegenerative disease at the earliest stages is an early deficit in regenerative capacity. That was our hypothesis. So how to test that? Well, there are ways of looking at structural plasticity where you make cortical windows and you look at behaving animals and you do super resolution microscopy and you can watch. But this is very intensive and very low, very high cost and technological and very low throughput. So we went to nature for sorry, I got the wrong way, for a model of synaptic plasticity, structural plasticity. So this is a hibernating small rodent. And it's well known in hibernation that two things happen. One is that, of course, you drop your metabolic rate and you cool down, your body temperature goes down. But in the brain, synapses are dismantled and then they reassemble on rewarming. And not only synapses, the whole dendritic arbor contracts on cooling and then reassembles, but it is known and established that this is a control synapse. You've got the PSD, the cleft, and a few vesicles. You can see on cooling that the PSD is dismantled and these vesicles are pooled. There is no transmission. And if you do an EEG on a hibernating rodent, it's essentially flat. There's no electrical activity. But rapidly and quickly, the PSD is reformed on rewarming. And you can see that now transmission has restored because the vesicle number. And you can follow this with mini EPPs and quantal bursts on neurophys. But this is a real, it's a phenomenon across the hibernating species. So we did an experiment. We said, okay, so if, so this is clearly a very important method for recovering brain function and recovering memories, because those squirrels, when they wake, remember where their food is. They remember where they've hidden their nuts. So this must be a carefully controlled process. So we first asked whether we wanted to test whether this is impaired in neurodegenerative disease and therefore in neurodegenerative disease mouse models. So we first had to do it in wild-type mice. So this is the work of Diego Peretti, who pioneered this entire methodology and program in the lab. So he took wild-type mice. This is a pseudo-coloured electron micrograph from CA1, showing you some representative synapses and a representative synapse density. He cooled them to the temperature of a small rodent hibernating for a few hours, and you can see there's a reduction in mean synapse number, and then he let them rewarm, and you can see this beautiful recovery of synapse number, and you can quantify that, and you can see this very robust recovery in synapse. So this is a big deal. We've done it in laboratory mice, but it shows you how conserved these pathways are. This is, mice don't normally hibernate, but they have the same process and the same response as hibernating species. So you can guess the next experiment, which is that we took 4 disease models, but I'm going to show you the data from these 2. We took the prion mice, and here we took the 5x AD mice, which Bart was talking to you about, which are a better model of synaptic loss than they are of neuronal loss. And we tested them in both of these, in each of these models. The synapse loss happens early before you get much, either UPR, misfolded protein accumulation, or even amyloid accumulation. It's sort of going hand in hand with those. And we tested them we tested their capacities for plasticity before they were losing synapses. So we did exactly the same to these mice. We cooled them, and here you can see prion mice very early in disease with a lovely plastic response here. But you can see just 2 weeks later, that recovery is eliminated. So they have lost the capacity to regenerate their synapses, and this is before the net synapse loss happens. Similarly, in the AD mice, at 2 months before they have any synapse loss, they have this very nice response. At 3 months, again before the net synapse loss begins, they've now lost that capacity very significantly. So in both of these models, the loss for structural plasticity precedes synapse loss. So this is the first, almost the first hit in a stress response that is going in these disorders. So this is the early deficit. So we wanted to know what the mechanism of this was. Now, the cold shock response is less known than the heat shock response, but it's another stress response. And it's well known that hibernation and hypothermia are neuroprotective. And everyone has their favorite story of someone who's been in a river at 2 degrees for 3 days and they recover and they have completely restored brain function. But they're well known, and it's used therapeutically in brain injury and in neonates with hypoxic injury. Hypothermia is protective, but the mechanism isn't known. But we do know that both of these induce downregulation not only of global protein synthesis and metabolism, except for one group of proteins which is upregulated, and these are the cold shock proteins. And the cold shock proteins are RNA chaperones. So they're interesting. The heat shock proteins are protein chaperones for refolding, but the cold shock proteins are RNA chaperones, so that you've got critical RNAs ready for translation once the shock is over, once the stress is over. So one of these is called RBM3, RNA binding motif 3, and this is a Western blot. I don't want to show you many of these, but just to show you that on cooling, you get a robust upregulation of RBM3 in the brains of wild-type mice. And in the prion and the AD mice, at that early stage, you get a lovely robust upregulation, but then that fails at the same time point as you fail to regenerate synapses. Okay, so that could be an association. But or it could be causal. So we asked if RBM3 was actually associated with the structural plasticity. So in other words, if we increase RBM3 levels, can we restore synapse regeneration in these models? So the thing to note about RBM3 is that the levels remain elevated for a long time after you cool. So we did this in 2 ways. We both did early cooling, boosted endogenous RBM3 by cooling these mice when they can still mount that response. So putting them basically in the fridge twice. These are mice with prion disease, which is a very rapidly neurodegenerative disease that destroys neurons within 12 weeks. The mice are dead with very marked signs. We cooled them twice, and we didn't cool the others, and then we just observed over time. And we noticed that the RBM3 levels remained high throughout the time course of normal disease. So what you can see here is synapses at a control mice at 7 weeks, synapse number, but the mice that were cooled but have the same level of prion disease have much higher numbers. And those are equivalent to wild-type mice that haven't got any disease. So these mice are completely protected from synapse loss by having been cooled early in their disease course. They've still got the same amount of prion protein replicating and associating, but they are protected from synapse loss. And that restores synaptic transmission. It restores behavioral deficits. This is a motivational task dependent on CA1 integrity, and you can see that the cooling, which is pink, has maintained baseline function, and this is memory, novel object recognition. But very strikingly, this is a control hippocampus. This is prion disease. You can see the extent of neurodegeneration here, and that's prion but cooled. So all you've done, there's no drugs here. You've just put them in the fridge. Slightly frivolous description of it, but basically, early cooling has provided profound neuroprotection, and this is as profound as all our other interventions genetically with PERK inhibitors, etc., in looking at the UPR. And this is also interesting. This is the survival curve. So here, all the animals are dead in 90 days. This is the effect of cooling of those mice. And here it's the outlier that's interesting. It can show you how potentially protective this can be. And this kind of increase is a very significant increase in a neurodegenerative condition. And of course, RNAi of RBM3 eliminates the effects of that early cooling. So it is a mediator. And the other thing we did, instead of cooling now, we just thought, right, can we actually do this by just putting our RBM3 in with lentiviruses? Can we? And this is the knockout as a control. And here you'll see that both the prion and the 5x AD mice here at 9 weeks, very synapse depleted, but those that have had an RBM3 injection before they develop disease or clinical disease are very protected at the level of synapse number, and again at the level of neuroprotection. And again, RNAi of that eliminates that protective effect and accelerates it. Okay, and again it restores synaptic function, it restores behavior and memory, and it's highly neuroprotective again, and you can see the effect of RNAi. So here we've got prion disease or Alzheimer's 5x AD. I haven't showed you the 5x AD pathology, but you can see that here, instead of cooling, we've now just put in the RBM3, and we've got that fantastic neuroprotection. Okay, so, and again, the virus on it. So this is focal injection into the hippocampus has also increased survival, where this is abolished or reduced by the RNAi. So that was a story of a new set of proteins that got the BBC very excited. There was a feature on this saying hibernating hints at dementia therapy, and you may all think, oh well, those mice were cooled to 16 degrees, it's irrelevant for humans. Bears hibernate at 33 or 34 degrees, and therapeutic hypothermia is 34 degrees. So the physiological temperatures at which you hibernate are different whether you're big or small. And bears have a diurnal, have a hibernome that expresses high levels of RBM3. So it seems a potentially relevant pathway. But of course, and it's a pathway for neuroprotection. But of course, cooling itself is an insult, really. So what we really want to do is to be able to interact at this level without having to cool. So the questions that we're looking at now are, what are the mediators of RBM3? Obviously, what's the relevance for human disease, and can we drive neuroprotection through RBM3 without cooling? Well, the mediators, we've made good progress on this. We worked in collaboration with Anne Willis and Mark Smales, who were working on coal shock for other reasons, and they had done a screen in cells which detected a new coal shock protein, RTN3. So this is RBM3 coming up SERP is another well-known RNA chaperone. This goes down, and RTN3 is an interesting protein because it's a reticulon protein. It's a negative regulator of base 1, and it has other membrane-forming and synapse-forming properties that are interesting. So it's a new cold shot protein, essentially. And we did an experiment where we put RBM3 virus into mouse brains, and we found that that brought up the RTN3. So it's being induced by the RTN3. So it's not just coming up in the screen and co-immunoprecipitating, but you can induce its expression with the RBM3. So as I said, it's a reticulin protein. It's an omega-shaped protein. It's involved in shaping dendritic spines and synapses. And there's some evidence to show its involvement in synaptogenesis and spinogenesis. It's a negative regulator of base 1. So we did the experiment. Instead of using RBM3, we're now putting RTN3, or the knockout, the siRNA, into mice. And basically, the RTN3 has the same effect as putting RBM3 in to the prion mice. You've got lovely protection, and that's reflected in survival and neuroprotection. The RNAi eliminates that, but also it eliminates the effects of cooling. So in fact, the RBM3 seems to be acting almost exclusively, or a very large amount of its effect is through the RTN3. So as you can see, if you RNAi the RTN3, then you lose the protective effect of cooling. So this is the pathway, therefore. RBM3, RTN3 neuroprotection. We would like to intervene here because cooling, although it's used therapeutically, it gives you pneumonia and it gives you blood clots, and it's certainly not practical as a dementia therapy. But if we could find a way to induce the pathway, and we're working very hard at that. So I said to you the relevance for human disease. So we don't know that RBM3 is, we didn't know that RBM3, whether it's expressed in human brains, whether it's detectable in human blood, etc. But we've been pioneering that. It is expressed in human brains. It's expressed in astrocytes and neurons. We can detect it in blood. My lab has pioneered the detection of RBM3 in blood. It relates to cooling. It goes up on cooling, and it relates to brain levels. And we've done that with a series of experiments. But we've got interesting collaborations on this with the neonatologists who do therapeutic hypothermia. This baby's being cooled to 33 degrees. We have seen RBM3 go up in their astrocytic and neuronal populations, and we can detect it in their peripheral blood. And babies respond to cooling by increasing the RBM3. So we're now looking to see whether we can use this as a predictor or even a biomarker of response. And the other group of people we're working with are these people. So when the BBC put out their dementia therapy, this guy here, Martin Pate, contacted me and said, I swim in cold water every day and I'm sure I get hypothermic. And I have 80 swimmers who've been taking their temperature and regularly get hypothermic, and we've detected RBM3 in their blood, and it goes up. So this is a messy study that won't be easily tailored for publication, but there's a nice story in it because they do have RBM3, and we will be doing some further studies with them in a much more controlled environment. But the main thing is we're also looking with Henrik Zetterberg at RBM3 in human CSF, and he has detected it. So we may be able to look at it as an early biomarker in Alzheimer's disease, or maybe as a predictor of response to certain therapies. So in terms of mechanisms of neuroprotection, the failure of structural plasticity is a real target for changing this curve and shifting the neuron. It's not a cure. It's not dealing with the A-beta load. It's not dealing with phospho-tau. But it does deal with neuronal vulnerability and the effects of those toxins. In a way, it resets the cellular phase or the failing homeostatic phase. It's a way of prolonging the defense phase to the insult. And as a physician, you're as much interested as just making people better as with actually completely solving the primary problem. So I think driving structural plasticity, as well as, of course, targeting the UPR, will be, what have I done here? Oh yeah, will be means of delaying neuronal loss. So I'm just going to thank my team. I don't know what my timing is here. Okay, I've got loads of time. but this is, so these are, everyone's changed, but this is what's been done at the MRC toxicology unit, where I still have a lab, and Cambridge, and is a really collaborative project between cold shock and translational experts and neuroscientists and clinicians, and it's been a lot of fun actually. But the real hero of this story is Diego Peretti, who's an Argentinian postdoc who's absolutely had the courage to pioneer this technique and spent a year counting synapses in a revision for Nature in 3D. That was the only thing we had to do, but it took him a year on his kitchen table. So he's been an amazing guy in all of this. And Hannah and Drew are pioneering the swimmers and the babies' work and the human RBM3 and the relevance in disease, and it's all heading into a much more translational phase, and everybody else is involved in other ways and in different projects. So thank you.